Hi everyone. Today's lecture is about antimicrobial resistance. So we've started talking about the antibiotics last week, and now we're going to talk about what the consequence was of these antibiotics and what we're seeing today that can be a real problem. So when we talk about antibiotics, the first thing I want to talk about is the spectrum of activity, and you'll hear these phrases used a lot as we talk about antibiotics. We have what are called broad spectrum drugs. These are antibiotics that are effective for more than one group of bacteria, meaning perhaps gram-negative and gram-positive, so they can wipe out a larger population of different types of species. When we talk about narrow spectrum drugs, these will actually target one specific group. For example, they may only target gram-negative bacteria, and so they only work on a specific population. And one of the ones that you should have already learned a little bit about is penicillin. This was the original narrow spectrum antibiotic that was used. And it was actually found to be susceptible, though, to microbial counterattacks, meaning some of them could survive with it. So over time, though, we've tried to alter or improve on the penicillins to make them harder for a bacteria to become resistant to. And what we're finding is that they can overcome some of the limitations of the original molecule. We can make them more complicated where a microbe may not be resistant to it as quickly. So this is just a diagram showing you some major classes of antibiotics, from the penicillins to the sulfonamides, tetracyclines, and how big of a spectrum they have in what they can control. For example, tetracyclines are very wide or broad spectrum, right? They have all of these gram-negative, gram-positive bacteria, including chlamydias, rickettsias. They can inhibit all of those. But we have some that are much smaller. For example, polymyxin here is only going to do a small population within the gram-negative bacteria, so they would be considered narrow spectrum bacteria. Hopefully that makes some sense for vocabulary. So as we start talking about antimicrobial resistance, remember this is the actual bacteria in the human host that would become resistant to the drug. So for example, if I take an antibiotic, I do not become resistant to the antibiotic, but perhaps some of the bacteria in my body would become resistant. So we're always talking about the bacteria having the resistance. So what is drug resistance? It's an actual adaptive response in which microorganisms are able to survive an amount of drug that we would normally think of as inhibitory. So we might give a normal dose of something and think that it would actually kill or inhibit a population of microorganisms. Drug resistance would be that it could actually grow at that level of drug. And because there's a lot of genetic versatility and adaptability within these microbial populations, it's very common to have antimicrobial resistance to these antibiotics. And it can be something that it can be intrinsic, which means the bacteria has always carried this gene of resistance, or it could be acquired from another bacteria that has resistance. One that is not resistant could acquire those genes. So how does drug resistance develop? It started as soon as we started using penicillin. So there was even it seen in 1940 when penicillin was first developed. Into the 1980s and 1990s, we began to see it in much larger scale. Part of that is probably the increase of antibiotics in the medical field. And what we're finding is that microbes become resistant to a drug when one of the two following things can happen. There's either a spontaneous mutation in one of its critical chromosomal genes that allows it to survive in the presence of the antibiotic, or it might pick up an entire new gene or set of genes from another species of bacteria. So remember, we have these trillions of bacteria in our body. Some of them may have resistance, and they may pass that on to another species. So that's where we start to see this horizontal transfer of drug resistance. So what does this look like, this chromosomal drug resistance? Usually what happens is we have a spontaneous random mutation. It may not even affect anything about the bacteria. The bacteria may not ever come in contact with the drug. But what might happen is that you get slight changes in its drug sensitivity to a certain drug, making it resistant. And there are things called persisters. These are able, in this case, the drug might slow or stop the metabolism, but the microbe isn't actually harmed by the antibiotic. Okay, so they can persist. So when the antibiotic is removed, you stop taking the dose, these bacteria then can come back in full force. So how do bacteria get resistance in a horizontal transfer? And this can happen in a number of different ways. So we call them resistant factors, and these would be on a plasmid. A plasmid is just a circular piece of DNA that contains some genes, and in this case we would be talking about one that contains antibiotic resistant genes. And what can happen is there's three different ways that a bacteria species that's not resistant could gain resistance. The first one is what is called through conjugation, which sounds exactly like what it is. It's cell to cell. Through the pilus, we looked at that. That was one of the appendages a bacteria could have. And basically it's just taking a gene and pushing it through that tube into the other bacteria. So it's a cell-to-cell transfer. Another way that it can happen is what's called transformation. And what happens here is, let's say you have an antibiotic resistant bacteria in your body, but somehow it dies and it releases its genetic material. Another bacteria can come along and pick that genetic material up, absorb it into it, and it becomes transformed by it. It has now got the genetic material of the bacteria that died. And then the last one is what is called transduction. So sometimes a virus can infect a bacteria, and when it leaves that bacteria to infect another bacteria, it will take part of the genetic code from the first bacteria, and it can actually transfer drug resistance that way. Okay, these plasmids that have drug resistance are naturally present in some microbes, even if they haven't been exposed to an antibiotic. It's just the sheer number of these bacteria, some of them are bound to carry a gene that makes them resistant to a particular antibiotic. And so it's actually the sharing of resistant genes that causes this rapid proliferation of drug-resistant species. So that's one of the biggest concerns, is that all of us are harboring probably some species that have antibiotic resistance, but it's that idea that they can be transferred to maybe a pathogenic, a bacteria that can cause disease, that those drug-resistant genes can be transferred to it that could cause a real problem where we would not be able to treat it in an infection. So when we talk about that, what we see is natural selection occurs when you have drug resistance. So what ends up happening is that a large population of microbes will always contain a few cells that have drug resistance. We talked about that. That's just by chance, random. And as long as there's not a drug that comes into that habitat, it doesn't matter. That probably gives no benefit to the bacteria species that has that gene. But if we bring in the drug that that particular population is resistant to, it is going to have a selective advantage, meaning that it will be able to reproduce where other bacteria will probably be killed by the drug. So what you get is this offspring that inherits the drug resistance. And if you kill off a number of the bacteria, the ones that are resistant will be able to repopulate that with more drug-resistant species. Okay, so what that looks like is, if you were looking in a lab, let's say that this is a plate that has these different colonies. All the white dots represent drugs that are, bacteria, bacteria that are not resistant to a particular antibiotic. And these two little red circles are drug resistant. So let's say it's penicillin, for example. If we took this and we added penicillin to it, what would happen is, after 1 hour, all of those white little cells, colonies, would die, right? Because they have no resistance. These two little red ones, though, would survive. And if we let this grow for 6 hours, it can repopulate that whole plate then with antibiotic-resistant bacteria. So you can see how quickly this could shift a microbiome if we introduce an antibiotic and you have some species that are resistant to it. So this is actually considered an urgent problem. The CDC in 2013 issued a threat report which said that it is potentially catastrophic. and that we may actually enter what is considered a post-antibiotic era, where even if we have antibiotics, they would not be able to treat these infections because the infections would not respond to it. So one of the problems we run into with this is that new and effective antibiotics are very slow to come to market. First of all, drug companies don't make a lot of money off of them. They're not economically lucrative. In addition, it's very expensive and time-consuming to develop these antibiotics, so there hasn't really been any urgency for the pharmaceutical companies. So when we look at it, it is a worldwide problem. Part of that is that we see other countries that do not regulate antibiotics may overuse antibiotics, and that happens in the US too. So in the US, we have nearly 200 million prescriptions written yearly. So think about that number of prescriptions of antibiotics. And the problem with this huge number is that 75% of these prescriptions are not for bacterial infections. They're generally for viral infections. In other words, they're being prescribed for something that they can't even treat because a virus is not affected by an antibiotic. But we're exposing all of our bacteria in our body then to those antibiotics. And that can increase that chance of drug resistance becoming a problem. So what has happened in the past is the physicians have often used a shotgun approach. They use a broad spectrum antimicrobial, which will treat a ton of the different microbes, right? Even what they call bystander. And what ends up happening is that we get a spread of resistance and often to pathogens, which means if you get an infection by a pathogen that is now antibiotic resistant, we have no way of treating it. And so this has led to what we call superinfections and adverse reactions. And so what the hope is that through education, we have a growing awareness that decreases this practice. And that takes place in two different ways. First of all, with physicians, not prescribing it to somebody who has a viral infection, or not prescribing it a broad spectrum if they can do a narrow spectrum antibiotic. But this also falls on the consumer, right? This means that when you go to the doctor and you've had a runny nose, a sore throat, and a cough, and the physician says, I think it's a viral infection, and I'd like you to wait a week or 2 weeks before you come back in, that we don't clamor at the door demanding that we get an antibiotic. So these are two different ways that we need to come at this issue. So it's one of the things that we're looking at. We do have issues, though, in the fact that we do have tons of excess of these antimicrobial drugs in the US, but we also see it worldwide. And a lot of places, including Latin America and Asia, these are not prescription medications. These are self-medicated over the counter. So we also need to have this global perspective on it. And drugs, when they're used in this way, are not only ineffectual, because often they're being used to treat a viral infection, but they lead to drug resistance. And now when you think of the world as how small it's gotten with the ability to travel, this drug resistance that we might see cropping up in Asia and Latin America can come to the US with that transfer then and have the same issues here. So it's one of those things that we need to look at, of making sure that we're treating antimicrobials with due respect and recognizing their limitations. So I hope this helped clear up some of that antimicrobial resistance, what that means. Always keep in mind it's the bacteria that's resistant to the drug and how that transfers. If you have any questions, remember, always email me. Thanks.